Okay, so I think I'll go ahead and get started. So my name is Steven. I'm a graduate student in the Lebo lab here at the Lois Pope Center. Our lab, one of the things that we do is we study neurotrauma and ways that we can essentially try to preserve synaptic integrity following injury. And so today I'm going to talk about sort of one of the main projects that I have been working on, which is looking at how glial-derived D-Serine contributes to synaptic damage after traumatic brain injury. So TBI is defined by the CDC as any sort of damage or blow to the head that is sufficient to disrupt the homeostasis of the head. And so in the US, there's approximately 1.7 million people who sustain a TBI annually. And a TBI can lead to a wide variety of symptoms of different severities based on the nature of the injury. So for example, from a cognitive perspective, someone may have confusion all the way up to loss of consciousness. People may experience personality changes such as irritability or depression. And they also might have different sort of sensation issues such as blurry vision or balance problems once they sort of have this TBI insult. And so a brain injury is usually broken down into two different phases. The primary injury phase is usually what occurs almost immediately to a couple minutes after the injury. This is due to the actual sheer mechanical damage that the brain is sustaining. So this results in things like blood vessel damage and blood-brain barrier compromise. We also have just gross tissue damage and some axonal shearing. A primary injury isn't really preventable besides either not engaging in some risk-taking activity or wearing a helmet. But what these primary injuries then lead to is this sort of secondary injury phase. And this is this cascade of things that can occur following the primary injury. So it can occur within hours to days, sometimes, persist through even weeks or months. And some of the facets that we see of the secondary injury are things like ischemia and edema. We have excessive glutamate release that can contribute to some cell death. In particular, what I'll talk about today is some synaptic damage, as well as some of the gliosis that occurs due to the injury, how some of these glial cells take on sort of a different phenotype compared to in a physiological state. And so the CDC a couple of years ago put out some statistics looking at TBI from the early 2000s to 2010. And essentially what they show is just, if we look around 2007 to 2008, in purple, the total amount of reported TBI cases seems to dramatically increase. We also have an increase that seems to correspond with total cases in terms of emergency department visits, so more people are actually addressing a TBI. And further, we have a decrease in the number of deaths here as shown in green. So given, like 13-ish years ago, there's been sort of this surge of awareness of TBI where people have, I think, changed their mindset from, oh, just walk it off to, hey, we should actually address this injury. This is something that can lead to different quality of life for patients for a long time. And I think this has continued to trend similarly now in this past decade, where you might even, for example, on the radio hear an advertisement about traumatic brain injury in, the aspect of a car accident, for example, and how it's important not to discount some of these injuries that, you don't see a brain injury like you might see a broken bone or a cut, but it's still important to address these injuries. And so another thing about TBI is that it's not only sort of broken down into a primary and injury phase, but sometimes also into a focal versus a diffuse injury. So when we think of a focal injury, it's kind of self-explanatory, where it seems to have a focal point, something like a penetrating injury, such as a gunshot. So within that focal point, we'll have a higher degree of sort of cell death and tissue loss, inflammation of peripheral immune cells, as well as gliosis. But if we think of the diffuse injury, which is usually more common in humans, this is oftentimes something like a blunt head injury, either motor vehicle accidents, sports injuries, just falling and hitting your head. And so this is usually more common compared to the focal injury. In reality, it makes more sense to lump together focal and diffuse injury. So for example, if somebody hits the front of their head, they're going to have more damage in the front than they will further in the back. And so one of the things that we're interested in, this sort of diffuse axonal injury, right, where we have this synaptic damage and axonal injury with glial activation, but we're not seeing any focal cell death. And so when we focus on this sort of glial activation, it's important to think about what we call the tripartite synapse, And so this is the idea that a synapse is composed of a presynaptic neuron, as shown here in green, a postsynaptic neuron, as shown in yellow, but also astrocytes that can ensheath this synaptic cleft. And these astrocytes are responsible for doing things like providing metabolic support. They may undergo spatial buffering, which is removing potassium from the synaptic cleft, and it allows the cells to fire another action potential. And one of the main facets that I'll talk about today is that these astrocytes also can play a role in regulating synaptic stability after injury. But oftentimes when we think of this tripartite synapse idea, we tend to discount the contribution of microglia. And so microglia have previously been thought of as sort of the resident immune cell in the CNS. But now we see that sort of over the past couple of years, we have a lot of evidence that's implicating microglia in more than just simple immune functions. So for example, during development, these microglia can help prune or form synapses. Even in postnatal animals, the microglia can serve to sort of remodel some of these circuits and synapses, and they can even contribute to network synchronization and local activity and things like that. So they have more of just an immune role, but they can also serve to modulate some of this synaptic activity that we're seeing. And so today, one of the main things I want to convince you about is how microglia are actually playing a very important role in synaptic dysfunction following injury. We hypothesize that microglia are creating and releasing D-Serine, which is then contributing to the TBI-induced hippocampal synaptic dysfunction that we observe. And I'll touch on D-Serine in a little bit as well. So when we think of some of the pathology that's occurring in terms of synaptic dysfunction with these microglia and astrocytes, the exact mechanism isn't completely known, but when we do think about this, one of the key players that comes to mind is calcium. And we know that calcium is largely mediated through NMDA receptors, particularly in the hippocampus. And this is sort of a double-edged sword when you think about calcium, because on one hand, if we allow enough calcium in, but not too much, we can then contribute to things that are beneficial, such as synaptic plasticity or long-term potentiation, as well as gene expression changes that sort of accompany that to promote that lasting plasticity. But if we allow too much calcium to enter the cell, we can actually then lead to things such as activation of caspases and sort of what we call this excitotoxic cell death, just because it's due to overexcitation and overactivation of these NMDA receptors that then leads to cell death. So this is often accompanied with a massive release of glutamate. But one of the things that we'll talk about today is sort of how we can have some lasting synaptic damage as well as it relates to D-Serine. And so when we think of these NMDA receptors, it's important to remember that we have three concurrent stimuli that are required in order to activate these receptors. The first is we need modest depolarization of the cell. And by doing that, we can remove this magnesium block, which normally sits in the pore. And that opens up the pore of the NMDA receptor to allow ions to flow in. We also have binding of glutamate to the primary agonist binding site. So glutamate is considered the primary agonist for these NMDA receptors. And then we also have binding of a coagonist such as glycine, or in the case of the hippocampus, D-Serine. And so D-Serine is actually the preferred NMDA receptor coagonist within the hippocampus. And so when we think of D-Serine, it's rather unusual because oftentimes we work with the L form of amino acids. So to see a D amino acid that has a physiological effect is rather unusual. And just to go over a quick biosynthesis of D-Serine, the astrocytes will uptake glucose from the bloodstream through their end feet. They then will metabolize the glucose into L-serine. The astrocytes shuttle the L-serine to the neurons, where the neurons form D-serine through the action of serine racemase. This will be an important enzyme that I'll talk about today. D-serine can then be released from neurons, and it can act on the NMDA receptors to elicit that action potential. And it can also be uptaken by glial cells, which have an enzyme known as D-amino acid oxidase. This is one of the primary degradation mechanisms for D-serine, so I'll also briefly touch upon it later. And then in particular, sort of the main crux of this talk is how D-serine is contributing to hippocampal synaptic loss after traumatic brain injury. So one of the first things that we wanted to do was identify under uninjured sham conditions, what is the function and expression of D-serine and serine racemase. So we performed a immunohistochemical study, as shown here on the left, and we found that serine racemase and D-Serine are both localized to these pyramidal cells, pyramidal neurons within the pyramidal cell layer. From a functional standpoint, we used different cell-specific knockout systems to identify the contribution of D-Serine to long-term potentiation of these CA3 Schaefer collaterals to the CA1 synapses. So we did this using both a neuronal knockout of serine racemase as well as an astrocyte knockout. So the neuronal knockout shown here in green is driven by a Phi1 promoter that drives the Cre recombinase to remove serine racemase. And what we found upon performing LTP studies was that high frequency stimulation does not elicit an LTP response in the neuronal knockout. However, we don't see any difference in the astrocyte knockout compared to the controls. So this was done in vivo, and taken together, it's showing that this neuronal serine racemase is actually very important for the normal synaptic transmission. Okay, but one of the things that we wanted to study is how is this system sort of changing after TBI? And the way that we model a diffuse injury in the lab is using the controlled cortical impact injury model. So again, the main reason that we're using this is that we're able to damage hippocampal synapses without causing any cell death. So this is a way that we can model this diffuse injury that we might see in patients. So for example, if we look at the number of new and positive cells within the hippocampus in the CA1, CA3, and dentate gyrus region, there's no significant difference. But using EM to analyze the number of synapses in the CA1 region, we actually see that there's a significant reduction following CCI. Again, modeling this sort of widespread damage that we have absent of cell death. And so we then examined, well, how is the expression profile of serine racemase changing following injury? So these images were done by a collaborator and provided courtesy of Dr. Derek Balu over at the McLean Hospital at Harvard. So we found again that in sham conditions, serine racemase takes that neuronal profile within the CA1 layer and some dentate gyrus, looking at GFAP and IBA1 as markers for astrocytes and microglia. They're expressed, but they're still kind of in this sort of resting quiescent level. By 3 days post-injury, there's a large change in the expression of serine racemase. So it seems to now be in other areas of the hippocampus and not so much as much in the pyramidal cell layer. We also see an accompanied increase in GFAP expression and IBA1 expression, suggesting that these cells are also undergoing this reactive phenotype. And when we look at a higher magnification, we see that for both the astrocytes and the microglia at 3 days post-injury, there's co-localization between their respective cell markers as well as serine racemase. And interestingly, this actually continues all the way through 7 days post-injury as well. So one of the things that I wanted to do was examine, using flow cytometry, sort of a more quantitative approach to see if we can corroborate these expression studies. And so we looked at two different things using flow cytometry. The first was we looked at the percent of serine racemase positive cells within a population. So for example, the percent of serine racemase positive microglia or astrocytes. We also looked at the accompanying mean fluorescent intensity, which is essentially an analysis of the amount of the protein they're expressing. So to what degree are they expressing serine racemase? So when we examined the microglia, we found that as early as 3 days post-injury, there was a significant increase in the percentage of microglia that were serine racemase positive. This persisted through 7 days. And we see a similar increase in the mean fluorescent intensity, suggesting that we have more cells that are expressing it, as well as at higher levels. Further, when we look at the astrocytes, at 3 days post-injury, they seem to trend upwards. There's no significance, but by 7 days, they do show a significant increase in the percent of serine racemase positive astrocytes, as well as the mean fluorescent intensity, right? So now we have this flow cytometry data that corroborates the expression data that we see here. But then we next wanted to ask, if we sort of move a little bit downstream of serine racemase, what are we seeing as well in the D-Serine expression? But so quickly here, the mRNA expression overall remains constant. But again, as I mentioned, we have different cell expression. And so it's important to try to parse out the different sort of results that we're seeing on the cellular level. But so as I mentioned regarding D-Serine, we wanted to see if this mimics the expression pattern of the protein as we would expect. And so again, in sham conditions, we see that D-serine is sort of present within this pyramidal cell layer. When we look at the molecular layer, as well as the stratum radiatum, which have a lot of these synapses of the Schaefer collaterals, we don't see as much labeling in sham conditions. And in fact, in a serine racemase knockout mouse in the CA1, we don't see any D-serine labeling at all. But when we look under injury conditions at 7 days post-injury, we see a similar effect where we have a reduction of labeling in the pyramidal cell layer, an increase of labeling in the molecular layer, which is evidenced down here. And so this is suggestive that now we have sort of a change in this profile, similar to serine racemase, where glia are now becoming expressors of the D-serine. So one of the next questions we had was, okay, to what extent are these glial cells contributing to sort of this D-serine release, and how much D-serine are they upregulating after injury? So what I did was I performed fluorescence activated cell sorting to isolate microglia or astrocytes from sham and 3-day post-injured hippocampus. We sent these to a collaborator, Dr. Herman Wolosker in Israel, to perform HPLC studies to examine the amount of D-serine and L-serine within the cells. And what we found was that by 3 days post-injury, within the microglia, there's a significant upregulation of D-serine. So again, this is actually sort of supported by the previous data looking at the protein expression. Within the astrocytes, there's no significant change occurring at 3 days. There's a larger degree of variability, but I still think it sort of matches up with the previous expression. And then when we look at something like L-serine, we see that there's no change across the board. There's a decrease a little bit in the microglia. I would presume this is due to the increase in D-serine expression that we're seeing. But we also have a larger overall amount of L-serine, and so it seems like the sort of overall difference isn't as large as with the D-serine. But the take-home message here is that now microglia, especially at least by 3 days, because we're looking to analyze 7 days as well, are upregulating this D-serine expression. They're also upregulating serine racemase expression. And so one of the questions we then had was, is there a behavioral sort of outcome difference that we see if we prevent D-serine or serine racemase within the microglia? And so in order to analyze learning and memory, we performed the behavioral paradigm fear conditioning. So just to briefly summate the fear conditioning paradigm, essentially we are trying to have the animal sort of learn that a neutral stimulus is associated with an aversive stimulus. In this case, our neutral stimulus is just a little box that we place the mouse into. The aversive stimulus is a shock. And the idea is that the animal should associate the box with the shock. And thus, after they've received the shock, they would sort of freeze up and elicit this freezing behavior, which would be indicative of learning and memory. And so the way that we perform this experiment is 5 days following our injury, we habituate the mouse in the box for 10 minutes. So they're able to just explore the box and run around. Following the habituation 1 day after, we perform the training, which is where we actually administer a 2-second long shock to the mouse. So they're able to hang out in the box and then they get shocked. And the idea is they should associate the box with this shock. And so on the subsequent day, at 7 days post-injury, when we perform testing, we would expect that if a mouse has undergone learning and memory, they would exhibit this freezing behavior. And so a higher freezing behavior would be indicative of sort of a functional learning and memory. So as I mentioned before, we previously looked at the neuronal knockout in sham and uninjured conditions, as well as the astrocyte knockout. And we actually extended that study to examine the contextual memory in astrocyte serine racemase knockout mice. So briefly, in the white and the black, we have our control mice, the sham in white and the injured in black. We see an injury-induced deficit in the percent freezing in our control mice. However, that injury-induced deficit is no longer present in the astrocyte serine racemase knockout mice, suggesting that the astrocyte D-serine and serine racemase is contributing to memory deficits. Okay, and so we then asked, is this sort of effect also seen in microglial knockouts? And so what we did was we again performed the fear conditioning, where in the white open squares, we have the control sham animals. In the black circles, we have the injured control mice. we see the injury-induced deficit as evidenced by a significant reduction in freezing. We then use two different microglial knockout mice that we have in-house. The first is a CX3-CR1 driven serine racemase knockout mouse in red, and the other is a TMEM119 driven knockout mouse in purple. And so again, similar to what we see with the astrocytes, there's no more injury-induced deficit that we're seeing when we compare the mutant sham to the mutant injured, suggesting that microglial serine racemase and D-Serine is also playing a role in some of this learning and memory deficits that we're seeing. And so the behavior is sort of this encapsulation of all these circuits coming together to have this phenotype and this outward behavior that we're seeing. But we then wanted to ask at a more sort of anatomical level, what are we also seeing in terms of synapses and dendritic spines? And so one thing we did was utilize Golgi staining to examine dendritic spine density and morphology. So the morphological criteria is adapted from a paper published in 2014 by Risher and colleagues. Essentially, they look at dendritic spines and determine the morphology based on the length and the width. So for the sake of this discussion, I'll just focus on mushroom spines and thin spines. The reason being that mushroom spines are these mature spines that are conducive to sort of synaptic plasticity, and they're considered these mature developed spines. The thin spines are sort of characteristic of degenerating spines that are often seen after a traumatic brain injury. And just to clarify the way that these are identified is that any spine with a width greater than 0.6 microns, so the widest width of the spine, would be considered a mushroom, so as shown here in blue. And then any spine with less than 0.6 microns width, but also less than 2 microns length is considered this thin spine. And so for the sake of the graphs that I'll be presenting, the way that they're presented is that one data point will represent one animal, but one animal is composed of 3 cells, and each cell is composed of 3 dendritic regions for a total of 9 dendritic regions per animal. And each region is at least 10 microns in length, so it's at least a 10 micron long dendritic segment. So what we first found was when we examined the spine density, we see that in our control mice, there's a injury-induced decrease in spine density occurring over that first week following injury, or at 7 days, we have a significant decrease in the amount of spines. In our microglial serine racemase knockout mouse, we no longer see that deficit. And in fact, we actually also no longer see that in the astrocyte knockout mouse, which is interesting because again, we see that there's benefits in the behavior for both of these cell types. So we then examined the morphology again, because we want to see the amount of mature spines compared to the amount of sort of degenerating spines. And as suspected, the injury induces a significant decrease in the percent of total spines that are mature mushroom shaped in the control mice. And this is no longer seen in the knockout mouse for either the microglia or the astrocytes. Sort of in contrast, when we look at the degenerating thin spines, we see an increase in the total amount of thin spines. This is again due to just the degeneration from the injury, that we no longer see these changes within the microglial or astrocytic knockout mice. So now this is suggesting that if we prevent serine racemase from the glial cell types, we're able to sort of protect these synapses from the injury-induced deficits that we're seeing. So I'd also like to say that I previously mentioned the TMEM119 mice as well. And those will be undergoing the Golgi staining as well. It's just awaiting completion in the meantime. And so since we're seeing these things where we have differential effects for different cells, if we knock out serine racemase in microglia, we still see effects even though we have serine racemase present in astrocytes. One of the questions that we're then now sort of honing in on is, to what degree are we having perhaps cell expression of different genes changed by the other cells? So for example, if we examine the microglial population in an astrocyte serine racemase knockout mouse, how does that compare to a control microglial population? And so the way we want to do this is using single cell RNA sequencing of the whole hippocampus at sham 3 and 7 days, where we can form a single cell suspension from the hippocampus, sequence the cells, and then determine the different gene expression of the cells and sort of cluster them into different groups. And again, the idea would be if we knock out astrocyte serine racemase, do we have different grouping and clustering of the microglia? And also, can we then further look at a specific cell type and parse that out into subpopulations? So there's been a decent amount of evidence now that microglial subpopulations may actually be contributing to different pathologies that we see after injury. And so we can not only look at sort of the global cell changes, but also the actual subpopulation cell changes as well. So all of this so far has been talking about, well, what happens if we remove serine racemase, right? So how, if we just ablate D-serine expression in general? But one of the questions we then had was, can we kind of take this a step farther and see if we can analyze something like D-serine release to see if we can mimic the functional improvements that we're seeing in the serine racemase knockout mice. So after doing some research, we find that D-serine, one of the main mechanisms of release is through D-serine transporters. Okay, and so there's two main D-serine transporters. One of them is SLC7A10, and the other is SLC1A4. So this SLC1A4 is considered to be sort of the primary glial transporter. They tend to prefer it for glial D-serine transport in physiological conditions. 7A10 is a little bit trickier in that in vitro studies have shown that neurons will utilize 7A10 for transport. However, there's actually recent RNA sequencing evidence showing that 7A10 is expressed within astrocytes in the hippocampus. There's also some IHC evidence. And so when we think about it, it really might be that both transporters are playing a role in the pathology we've seen, especially after a traumatic brain injury. And so we want to question, well, what's the contribution of these transporters to this pathology? And we hypothesize that the transporter-mediated glial D-serine release is leading to this hippocampal synaptic dysfunction following TBI. So we first wanted to examine the general expression of the two transporters within the whole hippocampus. So this is mRNA expression using qPCR. This is plotted as the log2 of the fold difference. So in this case, the dotted line indicates no change at all. Anything above is an increase in expression, and anything below is a decrease in expression. We find that as early as 1 day post-injury all the way through 7 days post-injury, the SLC1A4 mRNA is significantly upregulated. There's an acute downregulation in the 7A10 mRNA that by 3 and 7 days sort of begins to now level back out towards a more baseline sham level. So interestingly, if you remember, at the beginning I mentioned the primary degradation enzyme for D-serine being D-amino acid oxidase. So this is sort of this astrocytic enzyme that serves to degrade the D-serine. And we actually see that there's an acute downregulation at 1 day, and it's sort of like starts to restore by now 7 days post-injury. But so when we think about this at 1 day, even at 3 days where it still has a little bit of a reduction, this might be contributing to sort of a lack of degradation within glia, which is then now sort of increasing the total amount of D-serine. We're then also seeing an increase in a transporter, and so this may be playing a role in allowing D-serine to be released from these glial cell types, which then leads to the pathology that we see. And so in order to further focus on SLC1A4, we generated a conditional knockout mouse with the help of the UC Davis Knockout Mouse Project. So we purchased a founder mouse from them, which is an SLC1A4 floxed mice that also contains a LAC-Z insert. So what we first did was we bred it with a wild type mouse that allowed us to get germline transmission of our gene and our allele. And we are then able to use these mice, which have essentially one allele of this construct, to perform LAC-Z staining because they still have the LAC-Z reporter gene. And then for our actual experiments where we can perform the knockouts, we mate these animals with transgenic phosphoglycerate 1 flipase mice. And what that does is the flipase will remove anything between these FRT sites. That leaves us with our floxed mouse that we can then cross with the cell-specific Cree ERT2 mice we have in-house for astrocytes and microglia to examine cell-specific ablation in either cell type. But so as I mentioned, we can use these mice up here for lag-Z staining. And so one of the things we did was we wanted to identify the general expression profile of the transporter under sham conditions compared to 3 days and 7 days post-injury. So I will say that one of the caveats here is that we can't determine a specific cell type sort of beyond a reasonable doubt, because we only know that a dot is a cell that expresses the transporter, but we don't know what cell type. But it still gives us an overall idea as to the general expression profile that we see after injury. So if we look at sham, there's expression within the cerebellum, the cortex, a little bit in this striatal hypothalamic area. But if we look more closely within the hippocampus, we see that there's a lot of punctate expression outside of the pyramidal cell layer, such as in the stratum radiatum and down in this molecular layer here. There seems to be some sort of background expression within the pyramidal cell layer. I think, due to the fact that this pyramidal cell layer is also so dense with cells. But when we then look at the 3-day post-injury expression, where we have the injury cavity and then directly under the injury, the hippocampus, it seems that now we have a little bit more expression within these other regions, such as the stratum radiatum. But also, if we particularly look here at this sort of molecular layer, it seems that there's now a stronger expression compared to up here. And even at 7 days post-injury, it seems that this expression profile continues to hold up, where we have this stratum radiatum and molecular layer expression. And so maybe you remember that it seems to kind of also mimic the D-Serine expression that we saw previously, where in the stratum radiatum and molecular layer, we now see more expression of D-Serine following injury. And so we've also been able to use some of these mice to examine their learning and memory, again, with the fear conditioning paradigm. So this is still a work in progress. I wouldn't want to draw too many conclusions. We are in the process of obtaining more mice for the control conditions, as well as obtaining the astrocyte knockout mice. But in short, it seems that our control mice, as expected, exhibit this injury-induced deficit. The microglial transporter knockout no longer show this deficit. And so this would suggest that this microglial transporter is contributing to some of the learning and memory deficits that we see. But again, this is still in progress, and we need to increase the sample size as well as add some of the astrocyte knockouts and perform the Golgi staining for these mice. But so a lot of this now has been using genetic ablation of a transporter or of the actual protein to remove D-Serine in some way. And we wanted to see, can we complement our genetic approach using a more pharmacological approach? And so we were able to get our hand on 2 different pharmacological inhibitors. The first, as shown on the left, is L4-chlorophenylglycine. I may refer to it as L4-CpG or just L4. This is the SLC1A4 inhibitor, and we will be using it at a concentration of 250 micromolar for the duration of the talk. Sort of on the other hand, we have BMS 466442, which I'll just refer to as BMS. This is the SLC7A10 inhibitor, and we're using it at a concentration of 5 micromolar. So to determine these concentrations, we did it based on the IC50 that was published in the respective papers. But we also performed a dose-dependent response where we did a concentration that was 10 times lower as well as 10 times higher. And we found that these were the optimal concentrations that showed the best effect. So the way we administer these compounds is a little bit unusual as well, where we administer them directly to the ventricle that's opposite to the injury site, so the contralateral ventricle. And the way we do this is using an Alzette osmotic pump. So this is a pump that looks kind of like a pill capsule. It's maybe about 1.5 centimeters long. And within this capsule, there is a reservoir that's surrounded by a salt sleeve and a semi-permeable membrane. And so the idea is we can fill the reservoir with our target solution. We implant this subcutaneously between the shoulders of the animal. And then for the duration of either 7 or 14 days, depending on the model pump that we use, it can draw water from the animal and actually pump the solution into the brain. So the pump is connected to this catheter tubing, which has a brain infusion cannula. That cannula would again go into the contralateral side, into the ventricle. As you can see here, where we have the hole of the cannula that sits in the ventricle, and then that's able to continuously deliver our compound for whatever time point directly into the CSF, and that allows us to get sort of a more accurate delivery to the hippocampus. And so similar to the previous studies, what we do is following the infusion of these inhibitors, we analyze learning and memory, and then we also examine hippocampal spine density and morphology. So we first examined the fear conditioning behavior at 7 days post-injury, and we found that when we look at the sham animals, the administration of either compound doesn't seem to change their freezing response. There's no significant changes. When we examine again the sort of control animals of the vehicle administration, we do see, as expected, our injury deficit. So it's always important to establish the injury deficit, especially because we're introducing a pump to the sham animals. And interestingly, similar to some of the trends that we've seen before, such as with the transporter knockout and the serine racemase knockout, we are no longer seeing a injury-induced deficit when we administer the L4 or the BMS. And so we also wanted to extend one of our studies to look at a more long-term time point. And what we did was we administered L4 for a period of 14 days. We then allowed it to wash out of the animal for another 2 weeks. And then we tested the memory at 28 days. The idea being, if we protect these synapses in the short term, can we then have a lasting effect in the more longer term? And this is actually what we saw as well, where even at 28 days with administration of vehicle, the animals still have a significantly lower freezing response that is now increased when we administer the L4. So administration of L4 is able to sort of preserve or protect these spines, both in the short term and the long term, which is what I'll show you in a little bit. So again, looking at the 7-day post-injury data, when we examine the spine density, we see that there's, again, the injury-induced deficit that we no longer see in the inhibitor-treated groups. We then can look at the morphology and examine the differences as well, where, as you would expect, we continue to have our injury-induced deficit in a decrease of mature mushroom-shaped spines. Compared to the sham animal, the inhibitor-treated mice do seem to have a lower amount of total mushroom-shaped spines, but it's still significantly increased compared to the injured, suggesting that the sort of, again, amount of deficits that we're seeing can be protected by preventing some of this D-Serine transport. And similarly, when we look at the thin degrading spines, there's an increase in the injury controls that we see a decrease with administration of L4, and there's a slight decrease with BMS as well, although it's not statistically significant. But so the idea here is that perhaps we're still able to provide some sort of protection for the dendritic spines following the injury if we administer L4 or BMS. And so we also extended our data to look at 28 days, where here we see that the spine density is preserved again at 28 days with administration of L4. We see, again, similar effects with the morphology, where we have a preservation of mushroom-shaped spines compared to the vehicle. And we also have sort of a lack of thin spines compared to vehicle as well. So taken together, between serine racemase ablation, some transporter genetic ablation, even though that's still a work in progress, and then a pharmacological approach, all of this is really coming together to show that if we have glial D-serine release, it's contributing to memory deficits, to synaptic dysfunction, and if we can have ways to sort of prevent this release, we can significantly improve the outcome of these measures. And so I know that this is kind of a lot to take in, so I think if we sort of break it down into a more artistic schematic, perhaps it can make things a little bit more palatable. So when we look under sham conditions, we have the presynaptic neuron on the right, the postsynaptic neuron on the left, a microglia up top, and an astrocyte at the bottom. Essentially, what we have is the serine racemase is present in neurons. They turn L-serine into D-serine. This is then shuttled out through transporters and released along with glutamate that can act on NMDA receptors. That elicits an action potential that we see here. And then the excess D-serine can be taken up by the glial cell types and degraded, for example, by DAAO. But so what's occurring now in the injured state? And so we see that now within the presynaptic neuron, we actually no longer have serine racemase present. It is instead in the microglia and in the astrocytes. We see that at least at 3 days post-injury, we know that there's an increase in D-Serine within the microglia. And so what we're hypothesizing is now happening is that this buildup of this glial D-Serine is actually being released. It may contribute to activation of either extrasynaptic or perisynaptic NMDA receptors. And this might be one of the mechanisms that's leading to some of this synaptic loss that we're seeing. So some of the decrease in spine density perhaps may be due to some of this, this binding. But I don't want to say I have a mechanism for sure. It's still a hypothesis, but this is based on some of the studies that we've done, something that we think might be playing a role in some of this dysfunction and damage that we see after injury. And so just to kind of tie everything up, microglia and astrocytes express serine racemase at 3 and 7 days, and glial D-Serine contributes to memory deficits after injury. If we ablate serine racemase or do some sort of D-Serine transporter inhibition, we can protect spine density as well as the proportion of mature spines in the CA1 region after the injury. And we can also show improvements in learning and memory after injury as well. So some of our future plans, as I mentioned, are the single cell RNA sequencing. I kind of brushed over that pretty quickly because it's still something that's in its infancy. And it's kind of a lot to parse out with all the different cell-specific sort of results that we can see. And then we also want to examine the D-Serine levels in isolated cells at 7 days post-injury. So when I isolate those cells, I do treat them with transporter inhibitors, and that's a way to help retain the amount of D-Serine throughout the sort of lengthy isolation process. And then we also want to finish up the learning and memory as well as dendritic spine analyses in our SLC1A4 knockout mice. So with that, I'd like to give some acknowledgements, particularly to Dan. So, you know, he's been sort of my mentor here through all of this. He's helped with the experimental design, the data analysis, and just sort of giving me some, I guess, inspiration and motivation when we see some of these results, especially in some cases where, you know, it can be a little bit hard to interpret. Madeline as well for her help with designing some of these experiments and interpreting results. Dana, who's a new student in the lab, she's actually going to be working on the RNA sequencing aspect of this project. Maureen as well, who is interested in helping with some of the Golgi staining. Our technicians, Jose, Maria, and Magda for helping with the mouse husbandry and all the surgeries, as well as my dissertation committee for all of their input through my progress meetings. As the collaborators that we currently have, so as I mentioned, we're collaborating with Dr. Derek Balu at the McLean Hospital for some of the IHC studies, Herman Wolasker at the Israel Institute of Technology for the HPLC studies, and Pantelis, who's allowed me to graciously use his microscope for pretty much all of the imaging for Golgi staining that I've done in-house so far. I'd also like to thank Oliver at the Miami Flow Corps. So obviously he's helped with the flow cytometry and the facts. He's been tremendous help with designing the experiments, and the neuroscience graduate program for sort of giving us these opportunities to conduct research and present and disseminate our results to everybody here, as well as our funding resources. So if you have any questions, please let me know, and thank you guys all for taking the time to attend my seminar.
That was a very nice seminar. Congratulations on all the beautiful work. I have a question about brain penetrance. In this model, did the compounds have to be brain penetrant? The BMS compound looked quite large, and the L4 looked much better in terms of possibility of entering the brain.
Yeah, so in terms of the brain penetrance, so I'll go ahead and start with SLC1A4. So the L4-CpG. So this one, out of a couple different phenylglycine analogs, was shown to be the most potent. And since our lab has experience with some of the intraventricular infusions, we decided to do it directly to the ventricle because the plasma to brain ratio with systemic injection was about 0.13. So systemic injection of L4 is pretty poor to penetrate the brain. But since it is specific and we have that background with the pumps, we thought it was still sort of the best approach to use a more specific compound. And then the BMS, so I spoke with the corresponding author of the article that published the BMS structure and showing it was a selective inhibitor. And essentially he told us that if you do some sort of systemic injection, esterases in the blood will degrade the compound and it'll kind of just become inactive pretty quickly. And so those are the two reasons that we decided to go ahead and do the intraventricular infusion, so that we can just sort of circumvent these issues with the systemic administration.
Makes sense.
I think just to add a little bit onto that, just as a reminder, after TBI, the blood-brain barrier is pretty much open for the first couple of weeks. So, you know, it's, if this is the window that you need to block these synaptic defects, then you probably could get both of these components in there during the optimal window.
Yeah, that's a good point, that we definitely have the compromise after injury. So sort of a sham administration and an injured administration could be a little bit different. Are there any other questions or comments or anything? Okay, well, if not, then again, thank you guys for attending. I appreciate any other feedback that you may have. Please go ahead and just let me know. And I'm always willing to talk literally about any sort of science or any sort of experimental design. So please, I'm always willing to take input from people to sort of give us that outside perspective that we might not always have ourselves. Thank you, everybody. Take care.